Good morning, everyone. It is indeed my pleasure and a privilege to be able to stand here and present my research findings. Um, as it was pointed out, I had presented a part of this research at a conference in Rome a few months back. And since then, I had a graduate student who completed the research, and I have added those results as well. Since this is a new technology and of course we have a new audience, I thought it's a good platform for me to introduce this emerging technology that is called low temperature. It's a novel technology. It's just about coming, maybe 25 years old. And this is being increasingly used in agricultural arena. Seed safe horticultural crops has a wide range of applications in food safety as well. I have co-authors. This is Dr. Lam Dong. He's a PhD on turmeric. He was the first one to use plasma technology in my program. And then Mr. Sanathanam, he's a graduate student. He just completed MS on using low temperature plasma for improving microgreens seed. So we'll be presenting that research here. Then Dr. Naichambang is a plant pathologist, a colleague, and he has done some work on food safety, seed pathogens. Dr. Xu is a propulsion engineer at a different university, and he is an expert in developing the system. So he makes the instrument for us, and also his postdoc and grad students help our students use the instrument. So as the global community grapples with the pressuring challenges such as climate change, which is on us now, dwindling natural resources because of rapid urbanization and such, and a rapidly growing population which never stops, innovative solutions are needed more than ever to ensure that agriculture can sustainably meet future food needs. While artificial intelligence and machine learning to drones and precision agriculture are steadily transforming the face of farming, making it more sustainable, efficient, and environmentally friendly, there still exists a need to continue to innovate and explore chemical-free technologies to improve crop productivity and food safety. One such chemical-free green technology on the horizon is the non-thermal low-temperature atmospheric pressure plasma, which I will be talking a little bit more about. So what is this non-thermal low-temperature cold plasma or atmospheric pressure plasma, jet plasmas, and so on? This is now referred to as the fourth state of matter. We have solids, liquids, gases, and now this is a highly charged particles plasma, which is considered as the fourth state, which is a weakly ionized noble gas or even ambient air or elemental gases. It is comprised of positive and negative ions, electrons, excited neutral atoms, free radicals, molecules in the ground state and excited states, and also some UV photons. Plasma jets and corona jets are generated by subjecting noble gases, mainly helium and argon and also air, by splitting the gas by passing electric barriers. Plasmas are being increasingly used in several agriculture applications, including seed germination, that's priming the seeds, improving plant growth, mitigating microbial infections, enzymatic activations, inactivation, improving the cooking quality of rice varieties, starch modifications, and many others. Plasma was actually, it's about 100, 150 years old when Tesla was the first one who said that this is going to revolutionize biomedical field. So the applications began several years back for heel wounding, healing wounds and thrombosis and such applications in medical science. And now it is since about 25 years, this is being increasingly found to be useful in agriculture applications. Hence the term plasma agriculture. Okay. So as I said, this is a fourth state of matter. We have solids, liquids, gases, and then these are all charged particles that flow as plasma. So these are molecules, ions, electrons, and there's some heat as well in this. High heat plasmas, high vacuum plasmas are used for industrial applications. And low temperature or atmospheric pressure at room temperature plasmas are used for soft biomatter like plants and foods. So there are natural phenomena as well. Plasma is found in nature in the form of lightning, space weather, astrophysical jets powered by black holes, and also nebula that form new stars and such they give. Atoms that are disturbed and the electrons, when they jump their orbits, generally become charged, and plasmas are those charged particles. They have man-made plasmas that we use. We are now concerned with this plasma, where this person put a finger, we can feel it. It's not hot. Some of the plasma applications in agriculture include increased germination, yield, plasma-generated nitrogen fertilizers, reduced bacterial infections of leaves and fresh foods, reduce residues, pest and mycotoxin removals, and non-thermal pasteurization. We are now started on goat milk. This year, this student is working on that. So on several new platform, it's a new platform of technology for all kinds of people. So how do we generate plasma? It's a very simple technique. We take helium or air or even elemental gas like nitrogen and carbon, and at controlled flow rates, we let it pass through here, and there is a plasma generator, and this is where the electricity is applied and the jet comes out. Okay. So the way we pass the electricity, the gas is broken, is this contraption here. It has several tubes, glass quartz tubes. In the middle is the electrode that generates the electricity to direct current. And we pass the gas in the cylinder here. It could be helium, argon, air, or even nitrogen. And then as it passes through the electricity, it's passed, that's breaks up the gases into the atoms are broken, and the neutrons, electrons, protons, and all kinds are released, and these come out as jet. It's a fairly small jet. This is the actual jet we created. This is the plasma generator, and comes out so a few millimeters long. Okay. Like I said, this is still an emerging technology. Scaling up is still a big problem because scaling up needs a higher power. And when you increase the power, it increases the temperature and actually cooks the food. We have cooked some carrots and meat in our lab when we increase the power to make it big, more output. So our university is Alabama A&M University. I'm a professor here, and it's abbreviated AAMU. So we are involved. This is a huge $40 million project with about 12 universities and 2 industries involved, and funded by NSF and led by a space scientist, Gary Zank. And ours is a small team. I'm in charge of plasma agriculture, so agronomist. I have a food microbiologist and a plant microbiologist, pathologist, food engineer, physicist, and food biochemist. They are part of my team. And promoting plants. We are looking at promoting early sprouting in turmeric, improving plant growth, yield, and curcumin content of turmeric. Also looking at seed germination of crops. They're increasing growth, they're improving nutritional content, and so on. We are also looking at food safety, mainly salmonella on poultry meat and listeria on cold smoked ready-to-eat salmon. So I'll be presenting mostly our research on horticulture crop, turmeric, and also microgreens. Turmeric is known for its anti-cancer, anti-inflammatory, and anti-Alzheimer's disease, among many uses, and is trending as #1 herbal supplement in the USA, particularly in southern Alabama. It is tropical rhizomatous crop, and it does well along in the warm climates of the United States. However, due to restricted growing season or variety traits, harvested rhizomes tend to be immature and with lower levels of bioactive compounds, curcuminoids. So this is a 9 to 10 month crop in its environments, but when we try to adapt it to the US, we are getting like 7 months of growing season, which is restricting its production, and particularly accumulation of bioactive compounds, which are called curcuminoids. So we have this a lot of immature rhizomes that either do not sprout or rot, or they sprout very late, and it's causing some problems with poor establishment. So this project, we were thinking of working on seeds, but my PhD student, he had problem with turmeric. He came to my lab with some rotten rhizomes and said, "Doc, what should I do with this? They're not sprouting." So I told him, go to Dr. Shu's lab and treat with plasma and see what happens. So he made a small experiment, and I took them to the other university, treated them, planted, and within 3 weeks or so, they sprouted. So then we said, let's pursue this as part of your PhD. And Lam did extensive research on plasma applications. Okay. So he conducted several trials, greenhouse variation among turmeric varieties, and also we used about 6 different varieties of turmeric, and we used helium gas plasma exposed to 90 seconds. And we looked at time of sprouting and seedling growth in the greenhouse. So these are the plasma conditions. It was at 8 kilovolts pulse amplitude, kilohertz, 6 kilohertz pulse frequency, and 1 nanosecond pulse width. And it was, this is about approximately 70 nanopulses rise and fall. So this is the plasma coming out here. And then we developed using 3D printing. We developed this small brush-like thing. We embedded about 6 to 8 plasma jets to provide a uniform flow of plasma so we can treat the rhizomes. So this is Dr. Lam Dong. He's now assistant professor at Texas A&M. He did PhD here. We developed this ring here using plastic, the 3D printing first. And this is plasma, this is rhizome. And these are plasma jets embedded in this ring. Later, we came up with this. And then first experiment, if you see these seedlings here, they're sprouted, these are treated. Behind this here, pictures not very clear. They did not sprout untreated. Same thing with these sprouted. The back ones did not sprout. And if you see here, these are sprouted ones. So then we transplanted these into pots. Okay. So he looked at rate of sprouting. So we have different varieties. On the y-axis here, there are 3 different varieties. On the x-axis is the number of days to sprouting. The green bars are treated, and red bar is untreated. Okay, he used only one treatment for control, so we have same. It's about 48 days control untreated, whereas the treated, they, depending on the variety, they're spotted very early. Okay, range somewhere around 13 days to about 28, 30 days. The same thing here. Then the pictures of the plants, these are treated plants, they grew taller, and then untreated were short. And look here too, tall versus untreated here, short ones. And these graphs here show the rate of plant height, okay, as a function of days after planting. The solid lines are treated, and the broken lines are untreated. So you can see the differences here in the treated and the untreated. Same thing in this different varieties. Had different responses. That was the first revelation for us that the variety matters. Okay. So these are the differences in the 6 varieties here, 6, 1, 3, 6, 8 varieties. So Vietnamese varieties, and the difference in their treated and untreated, percentage increase in their germination, sprouting. Same thing here. You can see the amount of variation between the varieties. Then we did another controlled experiment where we harvested the rhizomes at 6 months after planting, 7 months, 8 months, and then 9 months. Abbreviated as 6 months old, 7 month, 8, 9-month-old rhizomes. We took one variety called CL9, which has high curcumin. And these are the treatments. If you notice, selected all uniform size rhizomes from each of the maturity groups. And what he did is he treated them. This is untreated control, treated for 60 seconds, 90 seconds, and 120 seconds. This is the days to sprouting. 6-month-old, the control never sprouted. The green bar is missing. The control just rotted in the pot, untreated. When we treat for 60, yellow bar, 90, red bar, 120 seconds, blue bar, the days to sprouting was very low here, only within 30, 31 days. The 90-second treated rhizomes all sprouted. And the sprouting was earlier depending upon the maturity group. So in the 7-month-old, 8-month-old, 9-month-old, the green bar, which is missing here in 6 months, this is the control. So it took about 2 months in the case of 7 months. And whereas this is 60 seconds, sprouted in about 44, 43 days. And the 90 seconds sprouted in about 28 days. And this is 120 seconds, about similar time. So 90 seconds, we found that optimal for sprouting very early. And days to sprouting also decreased with the maturity of the rhizome. So these are pictures from the greenhouse trial. The first row is 6-month-old rhizomes. This is control. The first horizontal row is the control. Second one is 6 months. Third is 9 months. And the fourth row is 120 seconds. So if you see here, whatever I showed earlier in the graph, we could see in the plants. This is the control, 6 months, never sprouted. They all rotted. And 60 seconds, they just started sprouting. Whereas if you see 8-month-old and 9-month-old, they already unfurled their first leaves. So there was a good correlation there with the maturity group as well as with the time of treatment. So just to give a quick highlight on this, comparing 6-month and 7-month, just 1 month made so much difference. These just rotted here in this control, whereas here you can see they're all coming out slowly, but they're sprouting in here. Okay. Then we transplanted them into the field to see if this plasma effects would continue in the field as well. And this is how we did. We plowed the field. We added a combination of cow manure, vermicompost, and poultry manure, and then made the beds covered with plastic. And then we transplanted the plants in the field, okay, and fed them with organic fertilizer. So in the field also, if you see here, this is the 6-month-old rhizomes and no rhizomes in the control, 60 seconds, 90 seconds, taller plants, more leaves, 120 seconds, little shorter than the tall one. It was consistent throughout. This is 7-month-old field, and you see the same kind of differences, 8 months, and this is 9-month-old. So we could see the plasma effects carry on from the greenhouse into the field as well. So looked at the plant height, similar trend, control 6 months, nothing there, but plants also grew taller. In the case of the plant height increased with the maturity group and also time of exposure, okay, and and also number of shoots per plant showed the same trend, increase with the age of the rhizomes. But then if you see the red bar, 90-second treatment was always much better, plant height as well as the number of shoots. So this is just to give you an idea in the field, how they look like for the number of shoots, more shoots in the treated ones compared to the untreated control. And then they are roughly, when you treat them with plasma, there is a difference of about 3 shoots per plant on average. So we took it to harvest, and these are harvested rhizomes. And look, again, yield was higher in the maturity groups. As the maturity group increased from 6 to 9 months, we got higher yields. And also treated, we got according to the treatment time. Again, 9 months gave exposure time of 90 seconds, generally gave higher yield per plant. Okay, very significant differences here. So I looked at the curcumin, which is the main bioactive compound. A lot of medicinal values of turmeric are attributed to curcuminoids. There's 6 curcuminoids. Out of that, 3 are in higher quantities and can be elicited easily. Okay. So first year we found a lot of variation effects of plasma. This was not consistent. We did following year again to see if plasma affects curcumin levels. Unfortunately, there's no effect on the curcuminoids. They were all over the place. So we feel that this gas, low temperature plasma, has more effects on the plant seed growth and growth traits rather than the quality aspects, the chemicals in the plant. Okay. Then with Lamb graduated and left, yeah, and then I got another student. His name is Shravan. He did an MS in my program, and he worked on mustard greens. So we started with mustard greens because NASA funded. They gave us some funds. Their rationale was that they're advancing their exploration in space, and they want to produce food in space. So they asked us what crop would be very good, because the problem there, they were saying extended stay in extraterrestrial environments, they lose focus. The astronauts lose focus, and food seems very bland. And they want something that would help the brain function, as well as something that will develop appetite for eating. Okay. So we did a lot of literature search in medical journals to see what kind of foods are good for the brain focus and so on. And we found that mustard greens are the best. They have isothiocyanates, which are known to be associated with brain functioning. A lot of medical journal papers have indicated that isothiocyanates in brassicas, broccoli and uh, all cabbages and mustards, and they have high levels of those, and they good for the brain. So we keep telling our children eat more brassicas. Um, so we looked at this. They have this can be grown as leafy greens, and they provide a lot of nutrients, very texture, flavor, and also mental health. Growing this in engineered environments, space habitats can increase abiotic stress because they have recycled water, which is used for many purposes on the spaceships, and they wanted to grow crops which have less water requirement. And mustard greens also are good drought tolerant crops. We wanted to see if plasma is going to help in all this. So we used two different sources of plasma, argon and helium gases, and we use the same levels here. First initial preliminary trial, we went all the way to 480 seconds of exposure times, these two gases, and we looked at the germination mainly in the petri dishes. Okay. Well, I'll come to these results later. Okay. So he did this, and we looked at their preliminary trial. We found that um, there is an interaction between gas type, argon or helium, with the time of exposure on the germination rates of the seedlings. Then we moved on to looking at different microgreens because they are high nutrient dense, nutrient and easy to grow. And their problem is germination can be patchy, it creates a lot of issues. And also they're grown in such high densities, which creates high temperature and relative humidity in the lower levels of the canopies of these microgreens, which is conducive for disease, pathogenic diseases. So there's dual problems of germination as well as disease. So why microgreens? These are all different varieties of microgreens that are out there. And these are all the health nutrients come out of it. And they have a lot of health benefits, anti-obesity to anti-cancer, inflammatory, and so on. And they are very easy to grow. Temperatures 18 to 24. They about 50 to 60%. And then seed densities are very high. Usually they're double what you use normally. And they do pretty good on all kinds of media. They can be grown in soilless media, artificial media, in aquaculture or hydroponics. They can be grown indoor, outdoor, all kinds of good. And they are very profitable. Production costs $5 to $8 per meter square, and they can make $20 to $25 per pound. See here, different types of media can be used and different ways of growing them. And these are some of the countries where microgreens are highly consumed. Darker the color, more the consumption. And it's a huge industry. It's now $335 billion industry, growing at 6.1%. A lot of big companies involved in microgreen production. So like I showed you earlier, this is how we generated the plasma. And this is a student. He looked at direct treatment of the seed and also a plasma activated water. We can treat the water and use that for growing these seeds and the seedlings. So preliminary trial, we had up to 480 seconds of treatments 4 replications. And if you look at this control here, seed germination was very high in the treated plants compared to the control. Okay, this is argon gas plasma, and this is helium gas plasma. So you can see the differences here. Germination time. Time to germination is key. Earlier they sprout and germinate. They have the early germination advantage, which translates into plant growth and so on. So these are different measurements he made, germination ratio, velocity, index, and so on. And all these measurements, we see that plasma treated were generally better compared to the control, and also varied with the time of exposure. Okay. So from this, we decided argon for 30-90 seconds increases germination percentage, growth rates, and also the vegetative growth index, and so on. And whereas helium treatments also showed some positive effects on germination. So we decided to hone in on this 30, 60, 90 second exposure times. And we did further work to look at the new shoot height. So there are some differences here, but and it varied with gas type and the time of exposure. So if this orange bars are helium, blue bars are argon. Helium tended to increase the shoot height compared to argon. And root length also, because we are looking at drought tolerance in mustard greens, and we wanted to see if this helps increase the root volume, which is an indicator of drought tolerance. Okay, so root growth was higher in this case, 90 seconds treatment. So biomass also was high. The notable thing here is that root index was root production, both argon and helium did pretty good, whereas argon alone was significantly higher in biomass production. And this is a very good combination of increasing the root volume without compromising on the biomass. Generally, when the plants make more root, it is at the expense of the shoot. But in this case, plasma is helping both biomass as well as the root volume. And same kind of studies where he used plasma activated water, and there are some effects here. Direct seed treatment is better compared to plasma activated water. There was some variation here depending on the gas type and the time of exposure. So all this in the, again, biomass. This is 30 seconds exposure, this is 60 seconds, and this is 90 seconds. So we had some variation here, gas versus the helium. Helium is this violet color, dark, and blue is argon gas. Okay. So plants again, pictures of the plants. This is control, a little shorter plants. The helium, a 30-second time of exposure, taller. Again, same with here, helium here. Plants look better in this. Okay. Then he did a nutrient composition. These are the procedures we used for sample preparation, and then looking at total phenolics, flavonoids, and also radical scavenging activity using two different assays, DPPH and ferric acid, FRAP. And here we found that we use plasma activated water and also direct seed treatment. And this is the control here. And this is for the growth rate, I believe. Yeah. And there's some variation there because of the plasma. These are direct seed treatments here. And this is plasma activated water. Okay. So again, looked at the seedling biomass. There are different experiments repeated to make sure that we are getting there. This is the total phenolic content. If you look here, and this is the control, and phenolic content exposure to plasma increased. Okay. Same here in this case. This is 60 seconds, water activated, plasma activated water. And this is activated water for 90 seconds. The way he did it is he activated plasma activated water, and then he soaked the seeds in that water for 30 minutes, and then he planted them. Okay. This is also on scavenging activity, FRAP. Again, light differences, not really great. This dark bar is control. These are treatments. And if you see here, there are some effects here, significant though. Same thing here, another one, FRAP measurements. This is 30 seconds, this is 60 seconds, and this is 90 seconds. The lines with bars with lines are helium, solid bars are argon. So you can see the light differences there. Okay. So again, flavonoid content showed some differences, but they were not statistically significant. Okay. So if you go back and see turmeric also, we didn't get good effects on the curcumin levels. Here, some of flavonoid content did not, but it's variable. In some of the experiments, we saw flavonoid content going up when we treated with plasma. Okay. So this is scavenging activity, DPPHSA. Again, some differences here, but not really great. So in conclusion, we found that helium-treated seeds, they outperformed argon-treated seeds across most parameters. Helium seed treatments combined with helium-activated water showed significant improvements in root length, biomass, antioxidant activity, particularly at the 60 and 90-second durations. Plasma-activated water had a substantial impact on germination and growth. Helium plasma-activated water outperformed argon. Plasma-activated water significantly enhancing final percentage growth, root length, shoot height, biomass, and also the scavenging activity. Significant interactions were there between the seed and water treatments, notably in root length, shoot height, biomass, antioxidant activities. And the combination of helium seed treatment and helium plasma activated water was most active. When you treat the seed and then soak them in plasma activated water, there's a synergistic effect. We saw even in turmeric, which I didn't present here, we had a high school student for summer, and I told her to do that experiment, and we found that there's a synergy in sprouting time. Same here. So then we also looked at all these different microgreens, about, I think we had 12 or so, and we looked at their imbibition. Seed germination, the first step is the imbibition of water. So when we treat plasma, seed is treated with plasma, the perforations are going up. We did one trial, no, I did not do it, but another scientist, another university, they looked pumpkin seeds. They are big seeds. They subjected to plasma and looked at the plasma seed coat under scanning electron microscope. I found that there are more perforations, holes on the seed coat, which is like scarification. It's a priming process and increases seed water imbibition by the seed, which kicks on the germination process. Okay. So we had radish here. This is blue line here is helium, 90 seconds. This is argon, 30 seconds control. So these radish, spinach, some of buckwheat and cilantro, they appear to be more responsive to helium gas, 90 seconds exposure. Here, cabbage, broccoli, pak choi, scallion, we thought maybe more argon. Okay. Sometimes not much difference, but here if you see broccoli, there's a big difference. The argon 30 seconds compared to control. So there were differences between the species, time of exposure, and the gas type. Okay. This is kale. No difference here between argon and helium. But fenugreek, no response. Control was much better than the treated. So this is after 24 hours of soaking in water. Some differences are there, some do not persist. And some crops we found difference, like buckwheat here, responded to helium 90 seconds. Cilantro in earlier one responded to helium, but 24 hours we found argon 30 seconds much better. So your time of exposure, time of soaking, everything is variable. Okay. Okay. And this is not just to give you an idea of seed safety. We looked at stem phylum botulism. It's a seed-borne disease on spinach. And we subjected that to the seeds were coated with the fungus, and then they were treated with or without plasma. So 4 days after treatment here, this is plasma is a blue line, and this control is orange. 4 days after treatment, we didn't see much effect in the mycelial growth. This is the vegetative growth of the fungus. And after 16 days, if you see here, mycelial growth in the control was very high compared to plasma treated. As we kept them in the pots, we in these petri dishes, the untreated, they produce conidia, which are propagules of this fungus. And somehow, in the case of plasma treated, there was no production of conidia. So it is having an effect on the reproductive phase of this fungus. Okay. Then this is campestris, Xanthomas campestris on pepper seeds. We coated them with this bacterium. And if you see here, this is control. This is scanning electron microscope plates. So the bacteria is a rod-shaped bacteria. It's pretty intact. But in this treated one, we find the bacteria is distorted, and they tend to conglomerate like this. And some of them, they're broken. The plasma destroyed the bacteria. You can see this one here. So the plasma membrane of the cell membrane broken. So this is complete death. Okay. This is on poultry. Poultry also, salmonella, we didn't see much difference. We treated them for 5 minutes and 10 minutes to argon gas plasma, helium gas plasma, and we didn't see much difference. Generally, for pathogens, and particularly food safety, you want a death of the organism because after 4 or 5 hours, they'll start growing back. So it can have some implications for its life. Okay. This is on poultry, and this is on listeria on cold smoked salmon, which is ready to eat. They don't treat this very well. These are treated cold treatments. So the listeria comes back on this. Okay. So see here, there's a reduction in the colony forming units of listeria monocytogenes. This is untreated 0. This is the 3 minutes, 6 minutes, 9 minutes, and 12 minutes. See the definite reduction. I think this is the plasma treatment. We use separate plasma instruments for microorganisms, food safety, and a separate one for seeds and such. So this is a different system called dielectric barrier. The electricity comes from these plates here, one here and one there. and the food is placed between them. And you can't touch the plates even when this is on. You could get shock, grounding effect. So in conclusion, plasma seems to have an effect on seed germination, turmeric sprouting, and the seedling growth, plant growth, and all. So we are thinking, how does it work? Plasma, particularly helium, produces hydroxyls and oxygen, hydrogen. And once they come out of the chute, they react with the atmospheric gases, particularly nitrogen, and they form all kinds of nitrogen molecules. And these are very transient. The one difficulty in quantifying the different chemical combinations and molecules that are formed, they move very fast because they're highly charged and they are still off their orbits, so they tend to stabilize. So they keep forming all kinds of compounds very quickly. That's one of the problems we are having in estimating what happens when the plasma impinges on the soft matter, what kind of reactions are occurring. So far it's very difficult to form, but speculations are that the combination of nitrogen molecules may be increasing the plant growth, photosynthesis and plant growth and such. And formation of hydrogen peroxide may be having antimicrobial effects. And also ultraviolet photons in this plasma, which also have sanitizing and disinfecting properties. So those may be some of the things that are happening that are causing. I am not sure. Yeah, I don't think we put that here. So this is all we have done. Is work is still progressing. We are now looking at increasing tissue culture plants of different crops using plasma, and also looking at goat milk, improving its shelf life, and also reducing the spoilage organisms in goat milk and so on. So it's an ongoing work. So that is the thing here, and I'm open to questions. This research has created a lot more questions than answers, but I'll be happy to answer with whatever we have. Thank you.